So I just got a new little pen pad, so we're going to test this out. And to do this, we'll talk about the lysosome storage disorders, or the lysosome storage diseases. So what are the lysosome storage diseases? Well, it's going to be a collective group of diseases where you're going to have a defective or a mutated enzyme that's missing. So the enzyme cannot perform normally, so therefore you're going to have an accumulation of stuff that cannot be broken down. And so the lysosome is going to be notorious for breaking down cellular debris. And it does that, and it also breaks down some extracellular organisms, so it's the breakdown center. So if you're going to have a lysosome storage disease, you're going to be missing an enzyme, which means you're going to have a buildup. You're going to have an accumulation of stuff within that cell. And there are going to be two main lysosome storage disorders that we're going to be talking about today. There's going to be the sphingolipidoses. Then also we've got the mucopolysaccharidoses. Mucopolysaccharidoses, and then the sphingolipidoses. So of the sphingolipidoses, there's going to be 5 main ones that I'm going to talk about today, and I'll keep them numbers just so we can keep them sorted. And then of the mucopolysaccharidoses, there's going to be 2 main disorders that we're going to talk about today. So in the sphingolipidoses, you're going to have an increase in sphingolipids. I bet that blew your mind. And then also, the mucopolysaccharidoses, you're going to have an increase in GAG, so the glycosaminoglycans within the cell, also known as GAG, so glycosaminoglycans. So we've got 2 different disorders, and now let's talk about some of the specific diseases. So of these, we have 5 diseases that are going to be categorized under the sphingolipidoses. We've got 2 diseases. So let's talk about this first one. We've got this first one, and it's going to be called Krabbe's disease. Krabbe. Probably named after somebody. Don't know who it is. But what it is you're going to have an enzyme deficiency, like I said. So we're going to have an increase in sphingolipids. So what sphingolipid are we going to be talking about? Well, you're going to have an increase in galactocerebroside. Alright, so you're going to have an increase in galactocerebroside. Then also, why do we get this increased in the sphingolipid galactocerebroside? Well, it's because we don't have the enzyme galactocerebroside ACE. So notice how the ACE ending, typically, just as a general rule of thumb, when you see ACE, it's going to break stuff down. And in this case, we're going to be breaking down this galactocerebroside within the lysosome into products. However, let's say that this enzyme is now taken out of the picture. We're going to have an increased galactocerebroside. So this is going to be Krabbe's disease. One kind of buzzword that I do want to cover is going to be gobloid cells. You're going to see cells that look gobloid. So if you're ever on a test and you hear gobloid cells, think Krabbe's disease, think galactocerebrosidase enzyme deficiency. So that's one of the 5. So now let's talk about a second disorder in that sphingolipidoses category. Because remember, up here we have the 2 different disorders. We've got the sphingolipidoses, mucopolysaccharidoses. Let's talk about our second disorder. And this one is a little more common. I would say Tay-Sachs disease. So we've got Tay-Sachs disease. And notice here that I do have a hyphen. The only reason I'm pointing this out is because later on I'll give you a little way to remember some of the major associations with these diseases. So Tay-Sachs is going to be a hyphenated name. So what are we missing here? Well, we're going to be missing the enzyme hexosaminidase. Let's see if I can spell this. Hexosaminidase. Alright. So this is going to be one of your major disorders. If I had to remember something, I would probably remember this one. Very testable material. So you're missing the enzyme hexosaminidase. What actually is going to build up then? What product is going to build up? Well, we've got GM sub 2 ganglioside. GM2 ganglioside is going to build up because this hexosaminidase, and this can get broken down into byproducts. Byproducts, you don't need to read it. This enzyme catalyzes this reaction, so all of a sudden you're missing this enzyme. You get a buildup of GM2 gangliosides, which are going to be a subtype of sphingolipid, because remember, these are still the sphingolipidoses. So let's talk about some more buzzwords. I love buzzwords. Onion skin. If you're ever told about a cell that looks like an onion skin or a lysosome, it's because you have this buildup of GM2 gangliosides. So if the lysosome appearance, okay, so onion skin, that's a big one. You're also going to have no hepatosplenomegaly. Hepatosplenomegaly. So you're not going to have an enlarged liver or spleen. We'll contrast this to a couple other disorders which do have the hepatosplenomegaly. So remember Tay-Sachs, think onion skins, hexosaminidase. I would commit this one to memory because that was an important enzyme. You're going to have a buildup of this product, the GM2 ganglioside, and it'll have an onion skin appearance to the cell. Alright, so now we've talked about two. Let's cover a third. Let me see if I can get rid of that. Okay, still learning the software. So now we've got a third disease. And this is probably one of my favorite diseases if I had to choose one. Still not a fan of these lysosome storage diseases, however, Niemann-Pick. Niemann-Pick. So, what do we know about Niemann-Pick disorder? Let's see if I can draw that hyphen in. Yep. So there is a hyphen in this disorder. And so there's going to be two hyphenated ones. Remember Tay-Sachs, remember Niemann-Pick, because at the end I will allude back to this. So Niemann-Pick disorder. What do we have? We've got an increase in sphingomyelin. Increase in sphingomyelin. Well, how in the world are we going to remember that? Because when you Niemann-Pick your nose, you need your sphinger. You need your sphinger to pick your nose. So that's how I remember it. That's at least some kind of fun way. So increase in sphingomyelin, which is going to be a sphingolipid breakdown. Except ours is going to be a sphingomyelin. So what enzyme, you may have guessed, remember you just throw ACE at the end and it'll probably break something down. So sphingomyelinase. There's our ACE ending. So that means we're going to break down that sphingomyelin into byproducts again. You miss this enzyme, you're not going to be able to break down the sphingomyelin. You're going to have an accumulation. So what's so different about Niemann-Pick disease? Well, if I had to remember, I would also throw that under that. Probably should remember this category. And then this one does have hepatosplenomegaly. Splenomegaly. So, if you have a patient that comes in with an enlarged liver and spleen, says you have probably a lysosomal breakdown disorder, you can rule out Tay-Sachs because that one doesn't. However, Niemann-Pick is going to be on your differential. Possible other answers as well, because, for example, Gaucher's disease has hepatosplenomegaly, but this is going to be one of the diseases that does have hepatosplenomegaly. So now let's cover a fourth one. I just said it. It's going to be Gaucher's disease. Gaucher's my bro. So Gaucher is going to be my awesome bro. It's going to be my... Oops, let's see if I can get rid of that. Man, I love that feature. Alright, we've got glucocerebrosidase. Bro. The bro disease. Gaucher's is the bro disease. There is the galactocerebrosidase. However, that's not as cool as Gaucher's. Gaucher's my bro. Alright, so we've got, this is the product that cannot be broken down into byproduct. And this is going to be catalyzed by the reaction that is going to be from glucocerebrosidase. So again, we're just adding the ACE to the end. Glucocerebrosidase enzyme. We're going to have that missing or mutated, so we have a loss of function. So we're going to have the increase in glucocerebrosidase. If I had to pick, I would pick these three disorders to remember. The Niemann-Pick one that we just talked about, number 3, and then also we talked about Tay-Sachs. Tay-Sachs, Niemann-Pick, and Gaucher's are going to be on my top 3 most important list. And I say that because there's the sphingomyelin, the onion skin appearance to the lysosome, the buzzwords. They've got buzzwords associated with them. And so Gaucher's has buzzwords associated with it too. This one is going to be Earl Meyer. My spelling is not very on, but Earl Meyer Flask. If you're watching this video, you're probably into the sciences. You probably know how to spell Earl Meyer Flask better than I do. Back in high school, I don't even know if I used a freaking Earl Meyer Flask. I like to space out in lab. That was kind of my study hall mental break. Alright, but back to this. We've got the Earl Meyer Flask lesions. So if in a clinical example they're giving you, or if you see in a patient, you're thinking Earl Meyer Flask. So we've got an Earl Meyer Flask, kind of looks like this, kind of got a little triangle down at the bottom with a long neck. So that's an Earl Meyer Flask to the best of my knowledge. You're going to see these in long bones. So let's say the femur of the leg. You're going to see these lesions, and you're going to say that they look like Flask-like lesions or Earl Meyer Flask-like lesions. You're going to be thinking Gaucher's disease, because this is a major association. So Earl Meyer Flask lesions, think Gaucher's disease. Also, another probably top 5, maybe top 3 takeaway message from this whole lecture is going to be the crumpled tissue paper appearance. Alright, what does that even mean? Well, remember, for here we had Tay-Sachs, we had an onion skin appearance to the lysosome. Well, for Gaucher's, we're going to have a crumpled tissue paper appearance. And for some reason, test writers love this. They'll either show you a picture of a crumpled tissue paper-like lysosome, or they'll just say it looks like crumpled tissue paper. I would remember this if I were you. The crumpled tissue paper is associated with Gaucher's disease. So we're still talking about the sphingolipidoses. So Gaucher's is just one of the 5 subtypes of sphingolipidoses. However, this one, the crumpled tissue paper appearance. And then lastly, I'm going to throw in hepatosplenomegaly. Just like the last one that I talked about, Niemann-Pick has hepatosplenomegaly, so does Gaucher's. Tay-Sachs does not. So that's 4 of the 5, so let's cover this last one. Here's our 5th, and this one's going to be Fabry's disease. Alright, so what do we know about this? Well, we know that it is going to be an increased accumulation of alpha galactosidase. My apologies, we're not going to have an increased one of this. This is going to be our enzyme. Oops, I just see if I can get rid of this. There we go. This is going to be our enzyme responsible for breaking down ceramide trihexoside. Struggling here with this pen. Into byproducts. this enzyme is going to catalyze that reaction. If it's missing, we'll have our increase in ceramide trihexidase. Alright, so Fabry's disease, not that important. However, I will point out that it is going to be X-linked recessive. So it is going to be one of our X-linked recessive disorders. When I say X-linked recessive, it means we're primarily going to see it on males. And then also I want you to associate one more disease with X-linked. So we have Fabry's disease. Then we're also going to have one of our mucopolysaccharidoses. So let's see if I can get back here. One of our mucopolysaccharidoses, there are two. And that one of them is going to be Hunter's disease. Hunter's. So Hunter's is also going to be X-linked recessive. Let's see, one more. X-linked recessive. So when I think X-linked recessive, I think Fabry's. I'm going to also think Hunter's. Everything else is going to be autosomal recessive. So X-linked recessive is going to be these two, the Fabry's. It's also going to be Hunter's, while our autosomal recessives are going to be for the other ones. So the Krabbe's disease, the Tay-Sachs, the Niemann-Picks, the Gauchers, and then also our very last one, which we'll talk about, is going to be Hurler's. But let's cover that now. So not much to say for buzzwords for that one. Now let's go into our mucopolysaccharidoses disorders. And of these, so if I go back here to screen 1, we're going to have two disorders. We're going to have Hunter's and we're going to have Hurler's disorder. So let's talk about our first one, which is going to be Hunter. Remember, I just said it's going to be X-linked recessive. That's an E. Hunter disorder. So what are we going to have? Well, remember we have the GAG breakdown, the glycosaminoglycan breakdown. For Hunter's disease, what we're going to have is we're going to have an increase in heparin sulfate, and then we're also going to have an increase in dermatan sulfate. Alright, so we're going to have an increase in heparin sulfate and also an increase in dermatan sulfate. So what enzyme is going to be responsible for this? It's going to be, oh, I spelled that wrong already. Let me find out how to erase it. There we go. This should be uronate sulfatase. Iduronate sulfatase. Iduronate sulfatase. IDURONATE sulfatase. Let's see if I can. No, that's about as good as it can get. So we're going to have this enzyme that's missing. We're going to have an increase in these products, which also we're going to have an increase in those products in our other mucopolysaccharidoses disorder. And really there's not much else to say about Hunter's disease. It's going to be lesser, more of like a mild disorder. However, we're going to have no corneal clouding. This is going to be how you kind of distinguish between this one. So no corneal clouding. So no corneal clouding. So then we've got our last lysosome storage disease. So that was one of two for the mucopolysaccharidoses. This is going to be our second one. This one's going to be, did I say Hunter or Hurler? That was Hunter, so now we're on Hurler disease. Hurler disease. It's an R. So let's talk about Hurler disease really quick. Also not much to say about it. However, so Hunter, there was no corneal clouding. For Hurler disease, there is corneal cloud. There is corneal clouding. So what builds up? Well, again, we're going to have the heparin sulfate and then the dermatan sulfate build up. So I'm not even going to write that again. Just remember, we're going to have the same byproducts build up. However, we're going to be missing a different enzyme for this one, This is going to be alpha L iduronidase. So alpha L iduronidase enzyme. Same products. This one's going to, present with developmental delays, corneal clouding, etc. So these are some of the basics for the lysosome storage diseases. So now let's see here. I promised earlier that remember the hyphen. So why are we going to remember the hyphen? Well, when you think hyphen, so Tay-Sachs and then also Niemann. When you think the Tay-Sachs disease and then the Niemann-Pick disease, because they both have a hyphen, think cherry red spot. So when you're looking in the eye through the fundoscopic exam, you're going to notice a cherry red spot, and this will help narrow down your differential diagnosis. So cherry red spot, this is probably another top 10 thing to remember for this lecture. If you hear the buzzword Ashkenazi Jew population, so the Ashkenazi Jew population, what are you going to be thinking? Well, you're going to be thinking of 3 diseases. You're going to be thinking Tay-Sachs, Niemann-Pick, and then to a lesser extent, Gaucher's disease. Probably Tay-Sachs would be number 1. Niemann-Pick could also be there, and then to a lesser degree, this would be my number 1 pick. So if I heard Ashkenazi Jew, I'm immediately thinking Tay-Sachs disease. You should be able to see a cherry red spot as well. That doesn't really help distinguish between these two, but it does let you eliminate Gaucher's disease. Like I said, you have Hunter's. Let's see. Just cross that out. Hunter's disease plus Fabry's disease. These are going to be your X recessive. All else is going to be autosomal recessive. Autosomal recessive for everything else. If you have any questions, let me know. I know I covered the basics and the buzzwords for a lot of these. I guess I could throw in that they're, let's see if I can get back to screen 1. So glycosaminoglycans. There's going to be 2 main groups. There's different types. So there's different types of glycosaminoglycans. However, you're going to have the heparin sulfate / heparin. Heparin versus heparan sulfate. This is going to be one type. You're also going to have a chondroitin or a dermatan sulfate, which is going to be a different one. So there's going to be a whole bunch of different types of glycosaminoglycans. However, our mucopolysaccharidoses, both the Hunters and the Hurlers, are going to be mostly the accumulation of the heparin sulfate and also the dermatan sulfate as well. So if you have any questions, let me know. If you have any video ideas, let me know as well. I'm always entertaining new ideas. Otherwise, please subscribe for more great videos or click like. I also enjoy your comments. Thank you very much.